Science Quest: Unlocking the Universe CRISPR Gene Editing. Imagine a world where inherited diseases like cystic fibrosis or sickle cell anemia are a thing of the past. Sounds like science fiction? Think again. Today, we're diving into the revolutionary world of CRISPR gene editing, a technology so precise, it's rewriting the very code of life. CRISPR-Cas9, to give it its full name, is essentially a highly advanced molecular scissor. Imagine the most precise pair of scissors imaginable, able to cut DNA at a specific location within the billions of base pairs that make up our genome. This scissor is a naturally occurring defense mechanism found in bacteria. Bacteria use CRISPR to snip out and disable the DNA of invading viruses, effectively creating an immune system. Scientists cleverly adapted this natural process to edit the genomes of other organisms, including humans. The guide RNA acts as the GPS, directing the Cas9 enzyme in the scissors to the exact spot in the DNA where the edit needs to be made. Let's break down the process with a simple analogy. Think of your DNA as a cookbook containing all the recipes for making you. If there's a typo in a recipe or genetic mutation, it can lead to a disease. CRISPR acts like a skilled chef with a perfect recipe book. It can precisely locate the error, cut it out, and then either replace it with the correct sequence or disable the faulty gene. This editing can be used to correct faulty genes that cause diseases, introduce beneficial traits into crops or animals, or even develop new therapies for genetic disorders. However, it's crucial to remember that this is a powerful tool, and we are still learning about its long-term effects. The potential applications of CRISPR are vast. Imagine treating genetic diseases like Huntington's disease, developing disease-resistant crops that require less pesticides, creating more efficient biofuels, even potentially enhancing human capabilities. But this power brings ethical dilemmas. We need to consider the potential for misuse, such as design of babies or unintended consequences of altering the human genome. Rigorous ethical guidelines and careful regulation are paramount to ensuring responsible development and use of this technology. What are your thoughts on the ethical implications of CRISPR? Let us know in the comments below. Currently, CRISPR is being tested in clinical trials for various diseases, showing promising results. However, challenges remain. Off-target effects, unintended edits in other parts of the genome are a major concern. Scientists are actively developing more precise and efficient CRISPR systems to minimize these risks. Another exciting area of research involves using CRISPR to target RNA instead of DNA. This allows for temporary gene editing, potentially offering a safer and more reversible approach to treating diseases. The future of CRISPR looks bright, but ongoing research is crucial to unlock its full potential safely and responsibly. While the science behind CRISPR might seem complex, its potential impact is undeniably tangible. Imagine a world with fewer genetic diseases, more sustainable agriculture, and innovative medical treatments. This is not just science fiction, it's the potential reality CRISPR offers. We need to continue investing in research, fostering open discussions about ethics, and ensuring responsible development to harness this incredible tool for the betterment of humanity. That's all for today's exploration of CRISPR gene editing. Next time on Science Quest, we'll delve into the fascinating world of fusion energy, the power source that could revolutionize our energy future. Stay tuned. Science Quest: Unlocking the Universe Episode 1: The CRISPR Revolution and Gene Editing. Did you know that scientists can now edit our genes with the precision of a word processor? It sounds like science fiction, but the reality of CRISPR-Cas9 gene editing is changing the world faster than we can imagine. Tonight on Science Quest, we delve into the revolutionary CRISPR technology, exploring its potential to cure diseases, enhance human capabilities, and even rewrite the code of life itself. Get ready to explore the future of genetics. CRISPR stands for clustered regularly interspaced short palindromic repeats. Sounds complicated, right? But the basic idea is elegantly simple. Imagine DNA as a vast library containing all the instructions for building and maintaining a living organism. CRISPR-Cas9 acts like a highly sophisticated pair of molecular scissors and a search engine combined. It targets specific DNA sequences. Think of it as finding a specific book in that library and then precisely cuts the DNA at that location. The Cas9 enzyme is the molecular scissor. Guided by a short RNA molecule like an address label, Cas9 finds the target DNA sequence and makes a clean break. Our cells naturally try to repair these breaks. Scientists can then use this repair process to either disable a faulty gene, effectively turning it off, or to insert a new, corrected gene, turning it on and replacing the malfunctioning one. The implications for medicine are astounding. Imagine a world without genetic diseases like cystic fibrosis, sickle cell anemia, or Huntington's disease. CRISPR offers the potential to correct the genetic defects responsible for these debilitating conditions. Clinical trials are already underway for various diseases, showing promising results in treating blood disorders, inherited blindness, and certain types of cancer. Think about it, a one-time gene edit could potentially cure a lifelong disease, eliminating the need for constant medication or painful treatments. This is a paradigm shift in medicine, moving us from managing symptoms to potentially eradicating the root cause of many inherited diseases. While the potential benefits are immense, the ethical considerations are equally important. The ability to edit the human germline, the genes passed down from generation to generation, raises profound questions. Modifying these genes could have unforeseen consequences for future generations. The possibility of designer babies engineered with specific traits sparks heated debate about eugenics and social equity. It's crucial to have open and honest discussions about the ethical boundaries of CRISPR technology and to establish strict regulatory frameworks to ensure responsible use. Science progresses faster than our ethical understanding. It is our collective responsibility to guide this powerful technology towards a beneficial future. The applications of CRISPR technology extend far beyond human medicine. Imagine using CRISPR to engineer crops that are resistant to diseases, pests, and harsh environmental conditions, thus improving global food security. Or consider the potential to eradicate disease-carrying insects, like mosquitoes, using gene drives, a technique that spreads genetic modifications through entire populations. CRISPR is a tool with immense potential, a key to unlocking a healthier, more sustainable, and more resilient future. But its power requires careful stewardship. We, as a society, must carefully navigate the ethical and practical challenges to ensure that this revolutionary technology benefits all of humanity. Today, we've explored the groundbreaking world of CRISPR-Cas9 gene editing, a technology poised to revolutionize medicine, agriculture, and beyond. Next time on Science Quest, we'll journey into the heart of the sun, exploring the latest breakthroughs in fusion energy. Will we finally unlock clean, limitless power? Join us to find out. Science Quest, unlocking the universe, a journey into CRISPR gene editing. Did you know that scientists can now edit our DNA with the precision of a word processor? Imagine correcting genetic errors responsible for diseases like cystic fibrosis or Huntington's disease, or even enhancing human capabilities. This isn't science fiction, it's the reality of CRISPR gene editing, a revolutionary technology that's rewriting the rules of biology. Today on Science Quest, we'll delve into the amazing world of CRISPR, exploring its potential and its ethical implications. CRISPR-Cas9, to give it its full name, is a gene editing tool derived from a natural defense mechanism found in bacteria. Think of it as the bacteria's immune system, but on a molecular level. Bacteria use CRISPR to remember and fight off viral infections. Scientists have cleverly harnessed this system to precisely target and modify specific DNA sequences in any organism, including humans. The CRISPR part refers to clustered regularly into spaced short palindromic repeats, essentially a unique DNA pattern that acts like a molecular address book. The Cas9 is an enzyme that acts like molecular scissors, cutting the DNA at the precise location specified by the CRISPR address. Once the DNA is cut, scientists can either disable a gene or insert a new one, effectively rewriting the genetic code. CRISPR potential is vast. Imagine a world where inherited diseases are eradicated. This is closer to reality than you might think. Clinical trials are already underway testing CRISPR therapies for various genetic disorders, including sickle cell anemia and beta thalassemia. But the applications extend far beyond human health. Scientists are using CRISPR to imagine rice that can withstand droughts or wheat resistant to fungal infections, enhancing food security worldwide. CRISPR could help modify algae or other organisms to produce biofuels more efficiently. CRISPR can be used to modify bacteria, making them susceptible to existing antibiotics again. With such immense power comes significant ethical responsibility. CRISPR raises profound questions about modifying genes in reproductive cells. Sperm and eggs could lead to heritable changes passed down through generations. This raises concerns about unintended consequences and the potential for designer babies. The cost of CRISPR therapies could exacerbate health disparities, making them accessible only to the wealthy. While CRISPR is highly precise, there's always a risk of unintended edits to other parts of the genome. Despite the challenges, the future of CRISPR is bright. Researchers are constantly improving the technology, making it more accurate and efficient. We can expect to see more clinical trials and a wider range of applications in the coming years. New techniques like base editing, which allows for single-letter changes in DNA without cutting the double helix, further refine the process and mitigate risks. Imagine a future where genetic diseases are a thing of the past, where we can engineer crops to feed a growing population, and where we can combat emerging threats like antibiotic resistance. CRISPR holds the key to unlocking a healthier, more sustainable future, but it's crucial that we proceed responsibly, guided by ethical considerations and public discourse. That was our deep dive into the fascinating world of CRISPR gene editing. Next time on Science Quest, we'll explore the mind-bending possibilities of quantum computing. Can quantum computers truly solve problems beyond the capabilities of even the most powerful supercomputers? Join us next week to find out. Science Quest: Unlocking the Secrets of the Universe, Episode 1: The CRISPR Revolution and Gene Editing. Ever wish you could edit your DNA like a word processor? Sound like science fiction? Think again. Today, we're diving headfirst into the incredible world of CRISPR-Cas9, a revolutionary gene editing technology that's rewriting the rules of biology and promising to cure diseases we once thought incurable. Get ready, because this is CRISPR Gene Editing Explained. At the heart of every living thing lies DNA, the instruction manual for life itself. This double helix, packed with genes, dictates everything from your eye color to your susceptibility to certain diseases. For decades, scientists dreamed of precisely altering this code, correcting errors, and even enhancing our capabilities. Enter CRISPR-Cas9. Imagine a highly precise molecular scissor. That's essentially what CRISPR is. It's a gene editing tool derived from a natural defense mechanism found in bacteria. The Cas9 part is an enzyme that acts like these molecular scissors, cutting the DNA at a specific location. The CRISPR part is a guide RNA molecule that directs Cas9 to the precise spot on the DNA where the edit needs to be made. It's like having a GPS system for our genes. Now let's explore some of the incredible applications of this technology. One of the most promising areas is treating genetic diseases. Think of cystic fibrosis, sickle cell anemia, or Huntington's disease, all caused by single gene mutations. CRISPR could potentially correct these mutations, offering a permanent cure. This isn't just theory anymore. Clinical trials are underway, showcasing promising results. But CRISPR potential extends far beyond treating disease. Imagine engineering crops resistant to pests and diseases, enhancing nutritional value and making agriculture more sustainable. We're talking about boosting food production to feed a growing global population. This technology could also revolutionize biofuel production and contribute to a more environmentally friendly energy future. However, like any powerful technology, CRISPR comes with ethical considerations. The ability to edit the human germline, meaning changes that can be passed down to future generations, raises profound questions. What are the long-term consequences of such edits? Who decides what constitutes a desirable genetic trait? These are complex societal issues that require careful consideration and open public discussion. The future of CRISPR is bright, but it's crucial to proceed responsibly. Strict regulations and ongoing research are vital to ensure this technology is used ethically and safely. Scientists are working hard to minimize off-target effects, unintended edits to the genome, and improve the precision of CRISPR. So, what have we learned today? CRISPR-Cas9 is a groundbreaking gene editing tool with immense potential to revolutionize medicine, agriculture, and beyond. While ethical considerations are paramount, the possibilities are truly breathtaking. It's a powerful reminder of the incredible advances happening in science, constantly shaping our future. Science Quest: Unlocking the Universe, CRISPR Gene Editing, a revolution in healthcare. Imagine a world where inherited diseases like cystic fibrosis or sickle cell anemia are things of the past, where cancer is treated with pinpoint accuracy, minimizing harmful side effects. This isn't science fiction, it's the promise of CRISPR-Cas9 gene editing, a technology so revolutionary it's rewriting the very code of life. Welcome to Science Quest. Hi everyone, and welcome to Science Quest. Today, we're diving deep into the groundbreaking world of CRISPR gene editing, a technology so powerful it's sparking ethical debates and reshaping the future of medicine. But what exactly is CRISPR? CRISPR, short for clustered regularly into spaced short palindromic repeats, is a revolutionary gene editing tool. It's essentially a molecular scissor, incredibly precise and efficient. Think of your DNA as a long instruction manual for your body. Sometimes there are typos, mutations in this manual that cause diseases. CRISPR allows us to go in and correct those typos, changing the instructions at a specific location within the DNA. The Cas9 part is an enzyme that acts like the molecular scissors, cutting the DNA at a specific location guided by a short RNA molecule, which acts like a GPS. CRISPR precision surpasses previous gene editing methods. Traditional gene therapy often involved inserting new genes randomly into the genome, potentially disrupting other essential functions. CRISPR, however, allows for targeted edits, deleting, inserting, or replacing specific DNA sequences with unprecedented accuracy. This precision is crucial for minimizing off-target effects, ensuring the safety and effectiveness of the treatment. The potential applications of CRISPR are vast and awe-inspiring. In medicine, it's showing promise in treating inherited diseases like sickle cell anemia, cystic fibrosis, and Huntington's disease. Clinical trials are underway, with some already showing remarkable success. Beyond treating diseases, CRISPR is being explored to develop disease-resistant crops, enhancing food security and sustainability. In cancer research, it's being used to modify immune cells to more effectively target and destroy cancer cells. While the potential benefits are immense, CRISPR power also raises significant ethical questions. The ability to alter the human germline, the genes passed down from generation to generation, raises concerns about unintended consequences and the potential for misuse. Discussions about responsible use, ethical guidelines, and potential societal impacts are crucial as we navigate this revolutionary technology. We need open dialogue to ensure that this powerful tool is used ethically and for the betterment of humanity. Research is constantly pushing the boundaries of CRISPR. Scientists are developing more precise and efficient versions of the technology, exploring new applications, and addressing ethical concerns. The future of CRISPR looks bright, with the potential to revolutionize healthcare, agriculture, and various other fields. Imagine a world where we can prevent and cure genetic diseases, enhance human capabilities, and create a more sustainable future. CRISPR could be the key to unlocking this future. CRISPR gene editing is undoubtedly one of the most significant scientific advancements of our time. Its potential to transform healthcare and improve human lives is undeniable, but it's crucial to proceed with caution, addressing the ethical implications and ensuring its responsible use. Next time on Science Quest, we'll explore the fascinating world of quantum computing, a technology that promises to revolutionize computation as we know it. Join us then. CRISPR gene editing explained, CRISPR-Cas9 gene therapy, genetic diseases, ethical considerations, CRISPR technology, future of medicine, genetic engineering, genome editing.